to glucose 1-phosphate. Oh, sorry, that's H. Just so there is no confusion, that is an H. So we go from glucose 6-phosphate to glucose 1-phosphate. And that's kind of our starting point for where it can now be shuttled off to make glycogen. And what's the next step is to UDP ribosylate it, we say. We bring in, UTP. We clean it up. UTP. And then we get out pyrophosphate, which is PIPI, and basically then water will actually hydrolyze that so we get 2 inorganic phosphates. That's part of the driving force. Let me see if this will, it won't let me do that. Okay. So we're going to have to draw this. So what does it make here? It makes the edge of my computer sucks. You get I'm just going to put U there for the uridine. So it has the diphosphate, so we would say that this is UDP ribosylated. And then now what you can do is you can use this to start to synthesize glycogen. And I imagine this would be phosphorylated because it's trapped in the cell. And then what leaves? UDP. And then we make now our alpha length 1,4. Like that. You're on the path to making glycogen precursor. So we can go down there and we can start to now build glycogen. Does that make sense? And so those are the ways that it can be diverted. It gets kind of complicated, right? But those are kind of the branch points. You think about glycolysis as the main highway that things want to travel down, but there are diverting pathways, state routes, side roads, where these intermediates, each one of these would be considered a metabolic intermediate, gets diverted if needed. What controls whether it's needed? Concentration of things. We talked about pentose phosphate here. Concentration of NAD plus is going to control whether things go through this pathway or not. If the NADP plus is low in concentration, what does that mean? It means NADPH is high. There isn't any free around, so we have plenty of NADPH, reduced cofactor, to do biosynthesis, reductive biosynthesis. So concentrations will shift and it will control the flow through these different metabolic pathways. It's not that one pathway is on, one's off. They're probably occurring simultaneously at different levels, dependent on the activity of enzymes and the concentration of things like NADP+. Does that make sense? I say that a lot. Does that make sense? I hope that doesn't annoy people. I just trying to figure things out. Okay, so if we want to keep going, right down here, step 4. Okay, we break the thing in two. I hear lots of whispers. Does anybody have any questions? Yeah, on the exam? What's going to be on the exam?
I like that question.
Okay. We talked about this. Going to basically another one of these. And then both of these can now go into step 5 here, step 6. And then step 6, right, brings in things like NAD plus, 2 of them, 2 PIs, and then out the other side we get 2 NADH, and then we're going to get 2 of the 1,3 phosphoglycerates. Step 7 is going to transfer those to H. So we have two ADPs, 2 ATPs out. Make the glycerate 3 phosphate. Okay, this is done by a kinase. Then we go down again, step 8. We don't do that because we're going to do a mutase. And mutase just moves things around. We have 2 of those, 2 of these. Enolase gets rid of that water. 2 waters leave, and we make the phosphoenol pyruvate, which is very high energy. Last step is the major one, right, that drives pretty much all this step 10 to ADPs. Brings that to ATPs. And we get now pyruvate. Alright. So we see those different pathways for this. We didn't do glycolysis, or we didn't do gluconeogenesis in class today. So only thing you have to remember about gluconeogenesis is one new structure. It takes 2 steps to get back to phosphoenol pyruvate, but it's a structure that we see in citric acid cycle. You take pyruvate and then you just carboxylate it, okay, to make oxaloacetate. Okay, this is done by pyruvate carboxylase, which basically takes CO2, because everything's 2, 2, 2. Does that. So this is pyruvate carboxylase. And then we go back here. The second step is GTP dependent. GTP, we use 2 of those. We've got 2 of these. We get 2 GDPs out, 2 CO2s, and we get back to that. Okay? So that step 10, remember, that has a lot of free energy. It is an irreversible process. Once it does this, it's not going to go back. To get it to go back, we have to bypass it with 2 enzymes, one intermediate, oxaloacetate. Highlight the gluconeogenesis pathway. And the last one's a carboxykinase. Okay. Phosphenol pyruvate carboxykinase. And everything else, remember we just reuse all the machinery, all the enzymes that catalyzed reactions that did not have a very, very negative free energy. Most of their delta G's at cellular conditions are near zero, so they're reversible. So gluconeogenesis. If we were in the liver, it can do the reverse. And all you do is just draw the arrows in reverse. H2O went in, or came out, H2O goes in. Okay. Here. Reverse all of that. And the H is going in. So we just redo all of that. Now here's the steps that we cannot reverse directly, right? Step 3, we use water with a phosphatase. And we take off that PI. Okay. So now we're getting some overlap. That's 3 used. And then if we want to go here, again it's a phosphatase. So we bring in water, PI leaves. And that's all there is. There's no new structures. It's just you have a different enzyme that's using water. So how can you kind of see what's going on now by seeing the structures? It's a web, yeah, but that's life. Life's messy. It's a web. Web of interactions. Okay. So that's how those pathways go along. Now let's talk about regulation. A key theme that we talked about is this idea of reciprocal regulation. At each step, there's at least one reciprocal regulation. Okay. Now, on top of that, there can be additional ones at very important steps. For instance, step 3 is very important because that determines whether things go through glycolysis or not. Step 10 is important because that determines whether the whole process is finished. So those steps actually have additional regulation on top of reciprocal regulation. So if we want to talk about this, let me get another slide for reciprocal regulation, because this, you can see it's good. So here's how we can think about regulation. Let's add another one. So let's talk about regulation of this. And let's just break it down. The regulation steps are glucose 6-phosphate, or glucose goes to glucose 6-phosphate. Okay, and like we said, delta G of this, very negative. Okay? And if we have the reverse process going on, okay, 2 is negative. So our first reciprocal regulation is controlled by the concentration of glucose 6-phosphate. On the hexokinase side, okay, so glucose 6-phosphate acts as an inhibitor. So I'm just going to do it this way. So high concentrations of this inhibit hexokinase. So it acts as what we call a substrate inhibitor. Now, in the other path, over here, okay, glucose 6-phosphate, okay, controls the rate because substrate concentration, as we learned last semester, controls the rate of a chemical reaction. And so it's a substrate level control for the phosphatase. So for the gluconeogenesis pathway, what I'm showing here is the same thing is reciprocally regulated. On this pathway, it inhibits, okay, glycolysis. High concentrations of this actually stimulate more to go through this pathway just based off Michaelis-Menten kinetics. And that's known as substrate level control. But you can see this idea of reciprocal regulation. Okay. Now, basically, the next step here, we go 2, and then 3, and then this third step is the next control step. This is where we get fructose 1,6 bisphosphate. Right? And so, okay, if we have glycolysis and gluconeogenesis in the liver going both ways, how do we regulate that step? Okay. Well, we said there are actually 2 ways, but one of them that is the reciprocal regulator based off of internal concentrations was AMP, ADP, right? So on this side, AMP/ADP can bind to, this is an allosteric molecule, and this increases the activity. If we go over here to the phosphatase, we said that ADP/, and I'm fine if you just lump them together, is a allosteric inhibitor of this type. So you can see the connection there. Green here means go. Okay? So between these 2, you can see on one hand, we see what activates one way, and it makes sense, right? If your AMP and ADP levels are getting high and they bind to it, that's telling the enzyme we need more ATP, right? They're getting high. So that's why that's a stimulator. On the other hand, we want to turn that off because we would like energy to be coming through glycolysis, and while we have that going, we want this pathway to be turned off so we can start to build up our ATP levels. Okay, let's just stick with this right now, and I'm going to go on. Let's just use internal controls for our example. Okay, so then we go through all the rest of these to step 10, basically, right? Step 10 is the next regulation point. Okay, in the reverse, of course, we have 2 steps for gluconeogenesis. Okay, then we get to pyruvate down. And what controls is reciprocally regulating both of these, acetyl-CoA. So on this hand, acetyl-CoA is inhibitory to this, okay? And on this pathway, acetyl-CoA is stimulatory to that enzyme. So now we can see these kinds of, the connections here. So that's right, if you boil it down to that, that's not too bad, right? Now there are additional things, right? That's the first layer. You see that there's always this reciprocal regulation. If one pathway is turned on, a molecule that stimulates that pathway turns off the other pathway. Okay? Now we have additional ones that we can also talk about and understand. And these are kind of feedback off the pathway or external driven kinds of processes. And I'm going to write this over here. So we have glucose 6-phosphate here. It can actually be diverted from here. And we have an enzyme that will divert this, and it will make not glucose 6-phosphate, but it will make glucose, not 1,6-bisphosphate, but it makes a different enzyme. It makes a different product. It makes glucose 2,6-bisphosphate, okay? And this enzyme that makes it is known as PFK2 to differentiate it from phosphofructokinase. Okay? Now, the activity of this, and I'm just going to draw what influences this, is external. Insulin. Signal. Okay. Insulin signaling activates that so that it will convert glucose 6-phosphate to glucose 2,6-bisphosphate. Now this acts as another allosteric regulator, okay? Over here, okay, this binds to phosphofructokinase and enhances its activity. It stimulates it. However, the phosphatase, it inhibits. So this is unique because external signals are now activating, okay, an enzyme that makes something that can inactivate one pathway but activate another. You see that? Now, what reverses this is actually glucagon. So this process can be reversed. So this reaction here can be reversed, okay, by a phosphatase, glucose 2,6-bisphosphatase, okay, and this is controlled, okay, by glucagon. And glucagon tells you, and we'll learn about this someday, that it says that the blood sugar levels are low. So you have, and remember, PFK2 and this glucose 2,6-bisphosphatase is the same enzyme. And what happens here is whether one's activated or not depends on whether it's phosphorylated or not. The phosphatase gets activated by phosphorylation. So glucagon causes phosphorylation. which activates the phosphatase domain. Insulin causes dephosphorylation, which causes the phosphofructokinase, or that's not phosphofructokinase, it's whatever, the glucose 6 P kinase to become activated. Isn't that kind of cool? So you have this enzyme that can do either function, but what it reads is whether you have an external signal or not. If you get high levels of glucose 2,6-phosphate, it stimulates phosphofructokinase, and at the same time inhibits the phosphatase, fructose 1,6-bisphosphatase. Okay? As you get more of this degraded, the concentrations go down. So what happens to the phosphofructokinase? It's not as active, more goes through the phosphatase pathway. Yep.
So does the fructose 2,6-bisphosphate act the same way that glucose 2,6-phosphate does?
Does it act the same
Like
way?
fructose 2,6-bisphosphate, does that act the same way?
Oh, this is supposed to be fructose. I screwed up. Sorry. It's fructose. Sorry about that. It's fructose. And it should be at the fructose 1 level. I'm sorry about that. So that should be after the isomerase. So here's isomerase fructose 6-phosphate, right? That's where it's working. Which, what does that do if you think about it? So is this okay? Sorry about that. I meant fructose, and yeah, this should have been that. So it's supposed to be fructose 2,6-bisphosphate. So now what does this enzyme do? Okay, this is kind of cool. We didn't talk about this. This enzyme, when there's lots of glucose present, what is it doing? It's siphoning off fructose 6-phosphate. It's lowering its concentration, making fructose 2,6-phosphate, which is going to what? Shift the equilibrium of glucose 6-phosphate more this way. So you're pulling more through multiple pathways. It's getting pulled out over here, just stimulating this, and that's going to start driving more and more through. Sorry about the flow. Here's fructose 2,6-bisphosphate. That one always gets me. Okay. Does that make sense now? We clarified that. Okay. So what we've seen here are reciprocal regulation at every step. Then as we start to get to these major steps, you start to see additional levels of regulation with fructose 2,6-bisphosphate, stimulating one pathway, inhibiting another. Okay. And things like additional ones for phosphofructokinase, things like ATP also can inhibit that. So as ATP increases, it can actually inactivate that and slow it down as well. So it has multiple regulatory factors there. Probably one of the more regulated ones are down here at pyruvate kinase. Okay. What are some things that we talked about pyruvate kinase also influencing it? We talked about fructose 1,6-bisphosphate, feed-forward activating it. Do y'all remember that? That this, the concentration of this, can feed forward. Concentrations are rising in the cell, and as it does, remember these are all in a, don't think about them in logical order. They're just banging around together. They're a mixture in the cytosol. So as this concentration starts to rise around, it's in contact with the pyruvate kinase. And it has binding sites that it will bind to it and make it more active. So this is an example that we talked about of feed forward activation. That concentration will enhance the downstream enzyme. And what will that do? Remember, this is the very negative step, so it will just start pulling more phosphenolpyruvate out to pyruvate. Lowers the concentration of phosphenolpyruvate. Now what does that do to every step up above it? Shifts the equilibrium, right? As that last step is yanking phosphenolpyruvate out, it's effectively pulling all the other substrates through the pathway via Le Chatelier's principle. Lowering that concentration, now your delta G says that everything else is going to want to reestablish equilibrium. That step pulls everything else through. It becomes more active. Does that make sense? Oh, you guys are stoked about metabolism. Excellent. All right, so that's a feed forward activation. We talked about in the liver that another thing that we have, right, we have here phosphorylation. Of pyruvate kinase, right, which can turn off the enzyme, and then of course a phosphatase can dephosphorylate it and turn it on. So that's basically a molecular switch. The pyruvate kinase via this can either be off or on in the liver. Off or on, depending on whether it's phosphorylated or not. Now, okay, this is kind of getting deep, but I like it. So, why do you need all the other things if just phosphorylation of the pyruvate kinase can turn it on or off? Why do you need feed-forward activation? Why do you need acetyl-CoA coming in and acting as an inhibitor? It's to tune the whole process. So if you've got a bunch of acetyl-CoA building up down here in the citric acid cycle, we don't need to send as much through to the mitochondria. So this feeds back and helps to fine-tune to make sure we're sending the right amount in. This is feed-forwarding to activate it more to help pull more and more through. So all of these different allosteric regulators helps to tune the process so it's not an all or none kind of thing. It has shades of activity that are related to the energetic needs of the cell. And so it's finely tuned by all these interactions. Does that make sense? Cool. I think that's deep. I like it. It's cool. All right. So those are the main ones that I wanted you to know for that one. Now over here, we saw one additional thing. We saw that, for instance, glucagon signaling. Glucagon signaling inhibits this. And what it does is it inhibits the production of the enzyme. It's not going to be expressed. Whereas, I screwed up. Insulin, right? Insulin says you have a bunch of this, why are you synthesizing? Insulin does this one. Glucagon in the liver signals that you should express that second enzyme. You should control the expression levels. So these are counteracting one another. And these are external signals controlling gene expression of the carboxykinase. So those were and if you go back to my slides, I laid it out like the 4 different tiers of regulation and how we see it all here. We see the substrate level inhibition. Or we see substrate level control at the last step of gluconeogenesis. We see allosteric feedback, inhibition, and activation. So allosteric. We see covalent modification of pyruvate kinase, so off or on based off of. And then we see gene regulation, control of the expression of proteins via external signals. So, is this too much, or are we doing okay? You're all so quiet. And then all, okay. I really apologize for that fructose part. I apologize. That should be fructose. 2,6-bisphosphate. Are you all okay there? So now you're experts of regulation of that. So start simple. What are the reciprocal regulations in each step? That's what we did, right? We started with the things that stimulate and then activate at each step. Then all we did was we went a step further and saw how external things can also do the reciprocal regulation, can influence that via this other enzyme pathway. Then we just added extra layers like pyruvate kinase, turning something on, and regulation of gene expression. Yes?
So what's an example of how you would ask this question?
What's an example?
Like, how would you say this?
Explain everything.
Okay. How would I ask a question about this?
Really?
That's a good question. I haven't had the exam yet.
How could we answer this in 50 minutes?
Well, I could easily ask you, something like, describe what reciprocal regulation is and how it is worked out in glycolysis. Now, that would mean that you would say, okay, at each step it would be the first layer like we just talked about. Okay, at step 1 we see that by glucose 6-phosphate. At step 3 we see this in 2 ways. We see it as AMP ADP levels stimulating phosphofructokinase and inhibiting the phosphatase, fructose 1,6-bisphosphate, phosphatase. We can see that as acetyl-CoA here. Does that make sense? Is that?
You'd like structures in words?
I wouldn't need you to draw. So here's the thing. I'm a structures guy. I would have you draw. I want you to know all the pathways and be able to draw them. But then I would ask word questions for the regulation. So I'm not going to have you draw out any of those molecules necessarily, but I want you to know what are the allosteric regulators and why would those be potentially good allosteric regulators. Does that make sense? So why the heck would AMP and ADP be used to activate phosphofructokinase? Well, it's because when they're high in concentration, that means the cell is low in ATP, and glycolysis is our ATP generating pathway. Does that make sense? So not only knowing where they're regulated, you have to know why they're regulated there. Negative delta Gs, right? We talked about that. That's the key concept. What regulate, so that they're reciprocally regulated. Why would you want that? So that when one pathway is on, the other pathway is off. And it works the best if there's a common molecule that can turn on one pathway and turn off the other, because they're both in the same space. Then the rest of that is why the specific allosteric regulators. For instance, acetyl-CoA here. Why? Because if we have high levels of acetyl-CoA, that means we're backed up in the citric acid cycle. We don't want to keep sending stuff down to the citric acid cycle. It's already backed up. So that can feed back and activate this, or it can also activate this because we might want to be pushing acetyl-CoA back up here so that it can be used in the synthesis of gluconeogenesis, for instance, because acetyl-CoA can be used to make oxaloacetate. Does that make sense? So that's why. Activate this. We've got plenty of stuff coming through down here. Well, let's use it to make glucose. Turn off. We've got everything backed up here either because we have plenty of energy coming through here or because, well, we want to make glucose in the liver. So we want to expend some of that energy. Does that make sense? Y'all okay with that? Am I overwhelming you yet? Okay, cool. All right. Is that helpful, or is that just more confusing? Okay, nobody wants to lie. Or tell the truth, so. Assume you're all liars. No.
Dr. Thomas,
Yes.
could you go over the regulation for the citric acid
Yeah,
cycle?
okay. And that's probably a little more simple. Okay, so, okay, we do this. Let's